whole body physiological shift as you get ready for birth. Birth is a very high activity event in your whole body. And I want to point out this little, I lost my pointer, but at 37 weeks, that's what we call the window for preterm birth. Oh, thank you. Down here. So any birth that happens prior to 37 weeks gestation, we consider preterm up until about 22 weeks, which is the point of fetal viability. And so I touched on tolerance earlier when I was talking about the second trimester, but I want to talk about tolerance really specifically here because I really do think it's the most interesting thing about pregnancy. Personally, this is what I study in the lab. And so this idea of tolerance is that the immune system is designed to recognize anything self versus foreign. So your immune cells are circulating in your blood, they're in your tissues, and they're going to interact with anything they come in contact with. And if it's yourself, they're going to check it and say, okay, we're good to go, this is fine. And if it's something foreign, they're going to say, oh, this should not be here, and they're going to try to remove it from your system. This gets a little more complicated in cases of autoimmunity, where your immune cells actually recognize self as foreign, or in some cancers where your immune cells recognize foreign as self and don't remove them. In pregnancy, though, it's inherently a foreign event. So your mom has her DNA and your dad has his DNA, but yourself, the baby, is a perfect 50-50 mixture of both. And so what this means is that the baby has DNA that's inherently foreign to mom. It's coming from the dad. And so the mom's immune system has to adapt and tolerate this fetus. It needs to know that these foreign materials that are inside of the womb are not actually foreign, and it needs to be okay with that and support the pregnancy. And a key driver behind this is the placenta. So the placenta is an transient extra-embryonic organ that develops in the first trimester of pregnancy. And it really is a jack of all trades organ. So it works as the fetal lung, the liver, the heart, pretty much most organ systems during gestation, up until birth when the baby's organs will start to function for the first time on their own. So the placenta resides inside the uterus with the fetus and is connected via the umbilical cord. And this large schematic here is basically if you zoom in on a cross section of the placenta. And it's separated really nicely into a few different layers. And I just want to touch on two here that we're going to talk about. So the first one are these little tree-looking blobs, and these are called placental villi, and they're comprised of what we call fetal trophoblasts. So these are fetal cells that are living in the placenta. They help form the placenta and give it the structure that it needs to do all of its functions. In this light pink reddish here is the maternal placenta. So these cells are derived from mom. And you can see here that there's in really close proximity to each other. Notably also, the placenta is really vascularized. So there's a lot of blood, and these fetal cells are in kind of bathed in this maternal blood in this middle ground sort of land. And if you zoom in even more, you can see that some fetal trophoblasts, where's my pointer? Oh. Some fetal trophoblasts actually expand from the placental villi into the maternal placenta. And so these trophoblasts experience direct cell-to-cell contact with maternal immune cells that are living in the placenta. And so because of this, maternal fetal tolerance is so critical so that these maternal immune cells don't recognize these fetal cells as foreign and try to get rid of them. One of the ways it does this is by actually altering maternal immune cell functions in the placenta. So I just have one example here that we know the most about so far, and that's natural killer cells. So these cells are pretty much everywhere else in your body. They're circulating in your blood, they're in your tissues, and their primary role is to destroy infected cells. But in the placenta, not only do they not destroy fetal cells, but they actually help support pregnancy. So these interactions are really important for what we call the revascularization of the uterus. And so this allows the baby to get blood from mom. So it's really critical that this healthy communication is established during pregnancy so that the fetus can continue to develop. And when there's a breakdown in tolerance, we call that immune dysfunction. So these are cases where mom's immune system recognizes baby as foreign, or baby's immune system activates and has complications with the mom. And I want to point out here that this is just one reason that women go into premature labor. There's tons and tons of reasons. a lot of which are maternal with things like gestational diabetes, high blood pressure, or what we call preeclampsia. But there's also a lot of environmental factors such as stress and alcohol use, and more socioeconomic factors such as healthcare access, race, ethnicity, and things along those lines. And the reason we care about this so much is that premature birth is a significant global problem. 1 in 10 babies in the US are born preterm. And you can see in this map that the US actually, and on this chart here, that the US actually has one of the highest rates of preterm birth. When we consider countries that we consider our healthcare system equal to, like Canada, most of the EU, Australia, you can see that there's a pretty stark disparity with maternal fetal healthcare in the US. And I just want to put some numbers on that because I think it's a little hard to conceptualize just looking at a map. This is from the March of Dimes for 2023, but in an average week here in the US, over 7,000 babies are born prematurely. And when you zoom in on Connecticut, about 65 babies a week are born preterm, most of which are happening at Yale New Haven Hospital. Yeah, of course. You can ask a question. Yeah. Mhm. Yeah, so it's definitely pretty split across racial lines. I think women of color and their infants are about twice as likely to succumb to maternal mortality or neonatal mortality. So which is definitely a significant proportion of preterm births, for sure. And one of, or I guess the impact of premature birth is that the baby is coming out prematurely. It's exactly how it sounds. So a lot of times these babies are underdeveloped, depending at what gestational age they're at, their organ systems aren't completely developed. And this can lead to all sorts of complications, such as lung complications, cardiovascular complications, neurologic, intestinal complications in the gut. Some of these can be mitigated pretty early on. They're short-term problems, such as respiratory distress syndrome. You can treat the baby with supplemental oxygen. You can provide ventilation for them, and usually they resolve pretty quickly. There are also a lot of long-term comorbidities associated with preterm birth, such as cerebral palsy, chronic lung disease, and these are issues that these babies and these kids, as they grow up, are going to have to deal with for the rest of their lives. And premature birth-related complications is actually the leading cause of death in children under 5. So it's really, really important that we continue to study this because there's over 1 million premature infants worldwide that are dying of these complications. And this chart kind of breaks it down in terms of neonatal deaths, which is deaths within 1 month of birth. And you can see that it's a pretty significant piece of this pie here. It's about 16%. Whereas kids who are dying at later ages, these are, kids that are raised like infants and toddlers, it's only about 1.1%. But when you compile these together, it is the leading cause of death in children under 5, which is why we're so interested in studying this and why our lab chooses to focus on this. And so this is just a little bit of schematic about what we do before I pass it off to Tyler to talk about some of his current research. But we're really interested in the maternal-fetal interface. I talked about natural killer cells in the placenta earlier, having a big role in maternal-fetal tolerance, but that's just one facet of the puzzle. There's a lot of other mechanisms by which maternal-fetal tolerance is established and maintained, and we're trying to get at some of the other details about that. We're also really interested in the development of fetal immunity and mucosal immunity in neonates, as we know that they're really susceptible to a lot of intestinal complications. And with that, I will pass it off to Tyler to talk about some of his current research.
Thank you, Kerri, and good morning, New Haven. Thank you all for coming out today. And please excuse our experiment in fetal and neonatal immunity back there, babbling. That's my son and my wife. Thanks for coming. I love you all. So we, in the Konyakova lab, there's several other members here too, and we'd love to really talk with you guys. I'm not gonna speak for very long because we want to leave space for conversation, and we want to hear from you too about your stories of your families, the pregnancies that many of you have carried, and just the blessing that's been, as well as the challenges, because we realize it's an incredibly unique time of life, and in some ways, we recognize that these quantitative studies are really important. But we also are strong believers in the qualitative things that are innumerable, but are incredibly important to be speaking about and sharing with one another. So we look forward to hearing from you. So I'll give a little brief vignette about one of the studies that we've done for a couple of years in the lab that brings another flavor of other things that we're interested about in development and maternal fetal immunity. And Kerri has brought a wonderful introduction about the placenta and its key role in an exchange of nutrients and oxygen from mom to baby. And another reason that I was drawn to this field in learning about the placenta is it's an incredible tool to study fetal development without ever having to touch a precious newborn baby. The placenta is, as Kerri mentioned, transient, and at the time of delivery, many of you know, is delivered after the baby, and many times is just discarded. But in the last few decades, we've learned some really incredible approaches to make use of this tissue to study fetal development that is, perhaps going to waste, but we've tried to basically take advantage of this. And in particular, Kerri introduced the idea that the placenta is derived from the baby. And so you can see here, this is a slice of a placenta, a term placenta actually. And I stained it with a variety of colorful antibodies. And you can see in red on the interior of these, each green circle is a villus, a villi, where this black space is where the maternal blood would be bathing the placenta and transferring all those oxygen and nutrients to the baby across this barrier. And on the inside, you have all these red immune cells right there, ready to gobble it up and receive what mom is transferring. Yes, sir, question. Yeah, you can repeat. Thank you.
Thank you for taking time for my question. My name is Donald Elder. I'm a local New Havian, if I can use that term. What's interesting to me is if the placenta provides immunity, is it something that can be captured or saved in a way that it can help a child when they're going through other kinds of traumatic or diseases or something like that?
Wonderful question. I hope the Zoom audience heard that too. Thanks for using the microphone. And yes, there's a lot of longstanding work from healthcare centers around the world where cells from the umbilical cord are saved and stored in a freezer for many years. And the idea is that theoretically, the stem cell potential of these fetal cells from early life could be used if necessary. And I'm not aware of placental cells being saved, particularly in this way therapeutically. It's a little more intensive to extract the number that are required. So these red cells are sort of seemingly very apparent here, but if we want to get them in a way where they're going to last a long time, it's quite tricky actually. And the other sort of very logistical problem that is experienced, whether or not you want to store cells from a placenta or an umbilical cord blood, is that to maintain that storage for decades and decades is actually very costly. And so to decide who bears the burden of that cost is very complicated when there's no way of knowing, will my child need these cells in 40 or 50 years or not? But wonderful question. Umbilical cord cells are often saved, and maybe it's a great experiment to do. If we saved some placental cells, might they be worthwhile down the road? Thank you. Any other questions? Jump in if you have a question. Yes.
Along the same lines, I was pregnant 17 years ago. And back then it was really trendy to create placenta pills that the mom would take. I just know it was very expensive and it sounded a little creepy to me at the time, but can you talk a little bit more about whether that makes sense and what would that be used for? Sounds like for the mom's benefit, not so much for the babies, but.
Yes, thank you for that question about placenta encapsulation. Very trendy. And our lab generally does not study sort of the use of placental encapsulation for postpartum reasons. And I think some of that stems from the fact that our clinical team are not postpartum caregivers. They mostly care for babies that are in the NICU rather than following up with mom, six weeks later. There's sort of mixed reviews out there. The scientific literature is not particularly deep about placenta encapsulation. It is very expensive. And we're not here to provide medical evidence, but basically I have to pass the buck a little bit and say, we don't study that particularly. And some people find it very valuable. Other people, are passing on that. But I'm happy I can talk more in depth later if you'd like. All right, so we're interested in the fetal side of the placenta, of course. And one of the questions that we've had for a while is, how does the fetal immune system allow for the baby to prepare and get ready to enter the world after birth? And a lot of you can appreciate that when you have a precious newborn child, they come out in the hospital and they're nice and rosy and they smell good and their skin is soft. But before long, after a few months, they're encountering an onslaught of germs and microbes and dirty things. And in reality, this sort of depicted dirtiness really actually happens immediately, even though we can't see it. The process of birth is rich with microbes that mom's birth canal has that are transferred to the baby immediately. And these microbes are then growing all over the skin and the gut of your child as they begin breastfeeding and drinking milk and having natural foods. And the preparedness for this exposure to the natural world is incredibly important. And so basically we ask the question, if we take cells from the placenta or other pediatric or neonatal tissues, and we do a molecular scavenger hunt where we look for traces of the microbial world at earlier time points, what might we see that's indicative of a preview of the natural world? And some colleagues of mine before I joined the lab did this study to look for traces of bacteria, fungi, viruses, foods, or human autoantigens. And in the study that was published a few years ago, Dr. Konyakova is our lab director. She's the boss. So this study published by her and our colleagues found that the in utero samples from small intestine, pediatric and infant small intestine samples were rich with bacterial molecules. And so these charts just depict different ways where they described the molecular profile of these samples, and we're really surprised to see this strong signature of bacteria in utero, which historically has been viewed as a completely sterile environment where there's nothing going on in there and there's no bacteria until after birth. So this was a first clue that perhaps there might be some way that mom is providing a preview to the baby during gestation that allows it to get ready for the big day. More recently, we decided to extend this study a little further. I remember reading this paper a few years ago before I got involved with the lab, and I thought, this is incredible. This is very, very unusual, and it's changing the way we think about pregnancy. So more recently, we've extended this approach a little bit differently and said, all right, instead of just looking for these small molecules, why don't we look for proteins, which are a slightly larger molecule? And we'll look beyond bacteria to a whole range of other microbial and interesting things from the outside world. And when we took placental cells and did this molecular scavenger hunt, we've recently seen across 5 different placentas this very unusual and striking signature of fungal protein. So a fungus is something like a mushroom or a yeast. You might bake bread with yeast. And we all have healthy fungi in our bodies naturally right now. And these fungi are just a part of normal life. You eat food, you're exposed to pets and dust, and we all have fungi. So we're really interested in digging in more specifically about, okay, these fungal molecules that we're seeing, are they indeed a way that baby is preparing to come out and defend against the environment that they will be exposed to after birth? But specifically, how is that happening? We have no idea. These findings are unpublished, and we actually have a Zoom call later today with a infectious disease doctor who's going to advise us and sort of share his opinion about what he thinks this might mean for pregnancy and for neonatal life after the baby is born. So in summary, we've shown you that pregnancy is a two-way communication system between mom and baby, where we have to be tolerant of a slightly different genome that the baby is carrying, and mom has to make sure to foster and take care of that baby by tolerating it. We've shown you that fetal immune cells are preparing for birth in advance, and that this might represent an immune education of sorts, like pre-kindergarten in a way, and get ready for the outside world. But there are tons and tons of open questions that this has left us with, and I often say to younger trainees that I get the chance to work with, science is far more about asking questions than it is finding answers. So really, whenever you have this idea like, oh, that doesn't make sense, I need to learn more about it, that's science. It's really not about saying these are fancy data or numbers. It's really about asking the right question. And so some of the questions we're thinking about is, how is this tolerance established and maintained? We really don't know exactly how babies and moms tolerate each other. It's an open question. How are these bacterial and fungal proteins gaining access to the womb in utero? No idea. How might this contribute to pathological preterm birth or support healthy birth? What are the consequences for the children as they grow up? If you're exposed to a virus or a bacteria in utero, does that make you more protected later in life from infection? Open question. And then lastly, how do we take all this knowledge and sort of integrate it to boost mom's immunity and baby's immunity during gestation and after birth? I highlight recently FDA approval and use of new vaccinations against RSV, respiratory syncytial virus, where just last year, or this year, for the first time ever, there are vaccines being offered to women prenatally that will protect their baby from RSV after they're born. So really important work being done about boosting immunity. And with that, we would love to talk more. Thank you so much for your time and attention. And I think, is Jennifer moderating the Q&A? We'll all just have a discussion together. Kalida will moderate the Q&A. And you can, what, you want to say any comments about yourself? And
Oh, yeah. So I recently joined the lab, and it's been great, working and learning about tolerance and how complicated it is. It's never like a one-way street. And I think it's such an important work because there's not a lot of people involved in reproductive health research compared to like other research areas. And so, yeah, it's really something that I'm glad that we get to share knowledge with community, allow people to understand like the importance of, being involved in research and aware of research. Yeah. And so I'm happy to take questions.
Thank you again. My question is, if we know that certain behavior in the mother degrades the viability of the placenta and certainly impacts the child, and we know that it is an extreme problem, urgent problem within the Black community, I'm wondering, do we have statistical data that lets us know when you engage in certain behaviors, the impact that that will have on the child yet to be born? Do we have any, how drinking or drugs, how that stress impacts the chances of that child being normal and some other kinds of things that impact our children at large?
With that, to piggyback, can I piggyback? Because with that, I really want to know, along with what he's asking, I'm trying to figure why over the past several decades in our community, mortality rates are super high in the Black community. I'm trying to get a grip on it because it seems that it doesn't fluctuate much, and almost never do we see a, decline. In fact, we're seeing inclines. And so I'm wondering, what's the relationship between the initiatives that Yale is doing, in the hospital with really attacking this challenge that seems to persist and increase?
All the research that's being done. Hi, my name is Florestine Taylor. I've been around New Haven for a good little while. That's the main reason I'm here, because we continue, Yale continues, does very good work, and et cetera. But then it seemed like the impact in the community, as the community is supposed to be targeted, is not declining. So what is going on with this relationship of this high mortality rate among Black women with their children? Something is not in sync. And I'm not going to try to blame the victims that Black women aren't, taking care of themselves better, but even women in fairly, different economic status, still something is going on that the rates seem to be continuing to increase.
Those are really good questions. I think in terms of like the specifics in New Haven and the data in terms of what Yale's doing to combat that, it's a really good thing, and I feel like I should probably look more into that, but I'm not completely sure what the numbers are, but definitely I think it's something that we're well aware, like on a national scale even, that like there is that disparity. I think I will say that a big part of that is also, prenatal care and, access to that, and also like health insurance and, people actually feeling like they can, people being able to afford to, go and receive all these cares. And that's like, sort of like a general thing, but I also don't know if you guys want to speak to that.
That went out to do breast cancer research, stationed all over the communities, within the lower income areas like South Genesee Street, what we call the Ville, New Hallville, the Hill. And still, and what bothers me most is we have the finest medical institution almost in the world sitting right here. And it bothers me that something's not addressed on a major level to impact the city.
Yeah, that's an excellent point. So the rate of premature birth hasn't really changed across the past decade at all, which is really surprising given how far our like medical like implementation has gone. I think it's really important to point out that a lot of times, especially now, there's a lot of initiatives that promote DEI, promote inclusion, but a lot of, and this is just like one tiny little aspect of it, but a lot of practicing physicians now, especially in reproductive care, because it is a lot less popular than a lot of the other specialties, and the population of physicians that are giving out and practicing reproductive care don't necessarily reflect the diversity of the community that they're serving. And so I think Yale has done a lot for, upstarting these DEI initiatives, trying to recruit more physicians of color, trying to recruit more female physicians. But I think a lot of the racial differences too come from, like Kalida alluded to, like socioeconomic differences and sort of systemic problems that, we know are a problem, but haven't really found a good solution to. And I think as, science continues to evolve, as research continues to evolve, Yale does a lot of initiatives to get younger kids from underrepresented minorities interested in STEM. As like the next generation of research scientists comes up, hopefully it will reflect the diversity of the population it seeks to serve. Because I think that's a big part of science. We're trying to solve all of these healthcare issues, but it's not a one-size-fits-all model. And we need to address that and look at that going forward.
Yeah, thank you. I really appreciate these very honest concerns. And I'll highlight one of our colleagues at Yale who's a obstetric provider, Dr. Tosin Adeyemo. We'll try to get this into the chat and share the link too, but she has a community program here that specifically tries to do this work. And she's a Nigerian physician scientist, and her passion really is extending this care into the community. And I talked with her a while ago. She's a good friend, and she had to hit the pause button on our studies together because she was also about to go on maternity leave herself. So I think she's been preoccupied, but I gladly sort of share with the women and men who are doing this work besides us. We're basically lab rats who are not really clinical providers, but there are a lot of wonderful people that we want to amplify and make sure that, if you want to talk to them, I can connect you personally and send an email and say, Dr. Tosin, can we have a conversation about this? And I know she and her team are incredibly dedicated to this kind of work also. Any other questions?
Hi, I know it's very early in your research, but I'm wondering about the fungal load in the placenta. How does that compare with fungal load in pediatric samples and adult samples? And also, are your samples all regional, local? And I'm wondering if there might be something to consider in the future, regional differences in the fungal load in the placenta.
Wonderful. Nerd by basic science question. I sounds like we have some scientists among us. Thank you for that question. The bacterial samples from the study that's published were from Pittsburgh. Our professor moved from Pittsburgh to Yale during COVID, and I think that study, those samples were from Pittsburgh. The 5 samples that have this fungal load were collected here in New Haven, locally. And this methodology is actually not absolutely quantitative, meaning it's unfair to try to compare the magnitude of the fungal sort of representation versus a pediatric sample. But we definitely are thinking about, now that we have this weird data, how do we design the next step study that makes that exact comparison? So we've talked about, swabbing the noses of babies or swabbing their skin or looking at their stool and making that comparison in a quantitative manner. So that's a great question. And we hope that our discussion today later with the ID doc will reveal that kind of angle. Yeah, the question was, do we think there will be a geographic variation? I think probably to a minor extent. I was reading a little bit about these fungi recently because I'm not an expert by any means, and I think there's a widely diverse range of these species that exist in nature, but they're also, a fungus is a fungus in some ways, so I think they will be similar and different. Yeah, but definitely across, especially if we cross an ocean and look in Europe or Africa or Australia. Yeah, great question. Yeah, please. We'll bring the microphone.
Oh, I'm sorry. So, a couple of slides back, I saw where herpes was at the top of a list. I guess that was the identifiable autoimmune or STD that has been detected within the placenta of a pregnant. Is that what I saw?
Probably.
Okay.
I'm not sure which
So does
slide
anybody
this is, but-
remember? Oh, it was the list of diseases off to the right.
Yeah.
And I saw herpes at the top, which was interesting to me
Yeah.
because I don't think I saw autoimmune diseases, but I didn't see HIV.
Mm.
And I know that, yeah, go back. Ah, okay.
Oh, hippies.
So do you know which
Yeah.
one's in orange letters?
Got
Do
it,
you
got
guys
it.
know what I'm talking about?
This one.
I
Hippies.
gotcha.
But
Why?
herpes viruses are, yeah, great question. Herpes viruses are a large family of viruses, and sort of there's many different flavors. The common herpes virus that might result in like a cold sore or a cutaneous little infection is sort of different from other types of herpes viruses, but there are a few that are known to be congenitally transmitted. If a mom has a serious herpes virus infection and gets pregnant, it can be transmitted to her baby. That's not what this study demonstrates, but there are plenty of other groups out there that study congenital transmission of viruses like herpes virus, or some of our colleagues at Yale studied COVID a few years ago, and they found encouragingly that by and large, almost all women who were COVID positive and got pregnant, they were not transmitting COVID to their babies. But that is not true of all viruses. There are certainly viruses that we know directly get transmitted. If you all remember in Brazil a few years ago, the Zika pandemic, where those are very scary instances where almost all women with Zika were transmitting to their babies with devastating consequences. So great question. Well, thank you so much. I think we can mingle and get to know each other a little further. Thanks again for your time.
Thank you so much to our guests. Please come on over and introduce yourselves. I know you mentioned some contact information. We can put that in the chat. Thank you all for coming out. Happy holidays. Our next breakfast will actually be on the second Thursday in January, and we'll be at the Peabody Museum. Check your email. We'll give you some details. Thank you.